The aim of our experiment is to study the different stages of mitosis in onion root tips using the acetocarmine staining technique. The material required are a dropper, glass slides, cover slips, forceps, watch glass, a beaker full of water to grow our roots in, filter paper to remove excess staining, N/10 hydrochloric acid, an onion, needle, surgical blade, burner, a compound microscope to observe our slide, Carnoy's fixative, aceto-alcohol, acetocarmine stain. Now let's start the procedure. Firstly, we will take an onion, cut its dry roots with the help of a surgical blade. Then dip the roots into a beaker filled with the water. And leave the onion dipped into the water for 3-4 days. New roots will be appeared soon. Cut the grown roots with the help of a surgical blade about 2 to 3 cm and dip them into the acetic acid and then fix it using the Carnoy's fixative. Leave it for about 30 minutes. The next step is take a watch glass and pour some drops of HCl on the roots. Now we will stain the roots with the help of acetocarmine stain. and after staining we will leave it for about 40 minutes. Now we will take the roots on a slide, and with the help of a surgical blade we will cut the darkly stained roots, and we will remove the excess staining by the filter paper. Now we will heat the slide on the burner. Moving to our next step. Add a drop of acetic acid and make a squash using a covered slip, and make sure that no air bubble is formed. Now we will observe our slide under the microscope. And this will be the result of our experiment. The various stages of mitosis, prophase, metaphase, anaphase and telophase can be observed clearly under the microscope. The first stage is prophase. In prophase, the chromatin begins to condense and the chromosome appears as thin threads and the nuclear envelope begins to dissolve. Second stage in the mitosis is metaphase. In this stage, the chromosomes align themselves over the metaphase plate, which is an imaginary line passing through the center of the cell where the chromosomes align themselves. The third stage is anaphase. In anaphase, the microtubules pull apart the kinetochores of sister chromatids towards the poles, and the chromosome assumes various shapes in this stage. The last stage of metaphase is telophase. In the telophase stage, the spindle apparatus disassembles, nuclear envelope starts to form again, and the chromosomes begins to uncoil themselves. After the metaphase, cytokinesis occurs. In cytokinesis, the cytoplasmic division occurs, then the cell divides into two daughter cells. During cytokinesis, there is a formation of cell plates in plant cells, whereas in animal cells, cytokinesis proceeds by formation of a cleavage furrow. Precautions: avoid under or over staining, avoid under or over squashing, avoid formation of any air bubble.